Let's talk a little bit about the parts of an experiment, and we'll use our brine shrimp lab as an example. Be sure to record this on the second page of the outline. There is a chart where you can record the parts of an experiment. First, we have the independent variable. The independent variable is the part of the experiment that you are changing or you adjust if you are the experimenter. Sometimes these get confusing. So a way to remember that is to think, if I am the experimenter, what am I changing? So think of the I for independent, the I for what I am changing. So this is a factor that you're going to test to see how it affects something else, what's called the dependent variable. In our brine shrimp lab, our independent variable, what we changed, was the salt concentration from one dish to the next. The independent variable is always recorded on the x-axis, or the bottom line, the horizontal line of our graph. So notice our x-axis here, the bottom line, that horizontal line, has our independent variable. The second part of an experiment would be our dependent variable. Now this will depend on our independent variable. So this is the part of the experiment that's measured or observed. Now when we say measured, we mean measured as a result of the change that we made, not a measurement we're making for our setup. What we're observing that happened as a result of our independent variable. So essentially it's the data we're collecting. What is the data that we are looking for in our experiment? In our brine shrimp lab, our dependent variable was the living brine shrimp that we counted after we allowed them to hatch. The dependent variable is graphed on the y-axis. So if we look at our graph here, we have the line on the side, or the vertical line. That is our dependent variable. Next is our control. The control is used for comparing. It's kind of like a baseline that we can compare back to. This is a setup that does not get that special treatment. So if we think of our independent variable as a special treatment, what we're changing and doing special for our setup, the control would not get that special treatment. The control in our brine shrimp lab was the 0% salt solution. It didn't get the special treatment of the salt or the varying salt concentrations that the other dishes received. Finally, we have our constants. Constants are all the other factors that are kept the same. So all the other factors that we want to make sure don't impact our results. In our brine shrimp lab, we have many, as we usually should. Maybe it would be the amount of water used when we mixed it with the salt, the type of water, the type of eggs, the number of eggs, the amount of time we waited, the location where we put all the Petri dishes. All of these are constants, and we could continue to list more that we kept the same, so we made sure they did not impact our results.